Okay, so good morning, everybody. It's my pleasure to welcome you to our today's single-cell omics lecture series, for which we have a very prestigious speaker today, Christoph Bock from Vienna. And before I come to introduce him to you, I would like to announce a few additional items. So the next talk will be given on the 22nd of April by Lala Hakverdi. She's from the BIMSB MDC in Berlin, and she will talk about reliable quantification of cell state velocities. So please join us again on the 22nd for this fabulous presentation by Lale. And then I would like to announce two events that are coming from Single Cell Omics Germany. There is a workshop that will be in Munich on September 12th to 13th this year on computational single cell genomics. The registration will come soon, so at the moment it's an announcement only, but please save the date and hope to see you in this in-person workshop in Munich, many of you. You see the Twitter link and the other links as well. Then the next slide, please. There's also a virtual workshop in which you can participate if you are interested in spatial transcriptomic data analysis in Python. This will take place on the May 23rd to 24th this year, and it will be mainly organized by the Thijs group from Munich. And there are a number of tools that are introduced, as you see here on these slides. For those of you who are interested, register now, please. Okay, and there are also still, I think, 1 or 2 slots free for speakers for this lecture series, where you're listening today to. So please apply now via this link that is shown here as a speaker. If you want to be present here and present your data and your work, you're welcome, more than welcome. Okay, now coming to the virtual lecture series of today and the presentation by Christoph Bock. Christoph I will introduce him in a minute. Before I come to this introduction, I would like to give you some information how this is organized. So Christoph will give a 30-minute presentation without any interruption or questions. But after that, we will have a 15 minutes moderated discussion. And please use your raise hand feature to indicate that you would like to ask a question, but also you can also write your question into the chat. I will read them and I will try to then ask you to give your question personally if you can do it. If not, I'll read out the question by myself later on. So these are the two options of asking questions. Now we will come to our prestigious guest today. So Christoph Bock is a long-known person working in functional genomics, starting with epigenomics and epigenetics and epigenomics a long time ago. We know each other for quite a while. And Christoph now is a very, very prestigious figure in the world of single-cell transcriptomics as well, and single-cell analysis. He has a very broad and very interesting definition of epigenetics, and I think he'll come to that a little bit together with single cell data. He's at the CeMM in Vienna, and there he's heading the genomics unit, not only for the CeMM but also for Vienna University Alliance. He is a bioinformatician who has contributed a lot of things to the bioinformatics world, but he is also one of these persons who's spanning between experimental work and bioinformatic work and going into clinical applications as well. So he has a broad spectrum of things he's interested in. And today he will give us an overview looking into the past and the future of cells, single cell analysis, epigenetic states in cancer and immunology. And it's my great pleasure, Christoph, to have you. And I'm very, very eager to listen to your presentation. Thank you very much for joining us and giving this presentation in the SCOG virtual lecture series. Thank you. And over to you now.
Good morning. It's a great pleasure to present here. I picked looking into the past and future of cells as the title of this talk. This is to be understood on a bit more long-term perspective than the RNA velocity that was already briefly mentioned. So if we want to know what a cell is doing in a few hours or has been doing a few hours ago, RNA velocity is a great tool. If we want to understand on a matter of days and weeks or even a lifetime how a cell becomes what it is and prepares for the future, the epigenome can provide us with this information. So when you think about the epigenome, a very common notion is the DNA methylation and the histone modifications as kind of the second code of the DNA, a layer of access control to the genome that essentially restricts a cell in terms of which genes it can activate at any point in time. And that's a very meaningful way of thinking about the epigenome. But today I want to take a bit of a more kind of higher level view that is very much built on this concept of the Waddington landscape, this idea that cells develop into what they are along kind of predictable and kind of epigenetically defined trajectories. What is very important in this regard is that it's not epigenetics or genetics or epigenetics or transcription regulation. It's really, these are two sides of the same coin, as was already kind of intuitively captured in the Waddington landscape, because underneath this landscape, essentially Waddington drew this, what we would today interpret as transcription factors, through kind of path, essentially transcription regulatory effects, keeping this landscape in shape. And then intuitively, it's also quite clear what happens in leukemia, for example. You mutate one of these transcription factors, the landscape changes as these ropes kind of rearrange. You get a differentiation block, and you cannot no longer go to the terminally differentiated state, or you entirely go off the chart and end up in some errant kind of non-physiological states, all of this related to diseases. Now, this is a bit of an intuitive view. Kind of fittingly, Waddington was almost a philosopher of nature, and certainly not a molecular biologist in today's understanding. But thanks to the rapid development of technology, we are approaching the point that we can take the Waddington landscape as something on which we can do mathematical calculations, data-driven predictions of how cells become what they are, how they make decisions at any point in time, and even calculating back how cells came to what they are by looking into their epigenetic history. This is really the perspective for today. I will look into kind of the fundamental biology of single-cell epigenetic states. I will describe some of these the applications of this epigenetic perspective to precision medicine, and then the last bit is really into trying to get into function and causality through using high throughput methods. First, start into the topic of single-cell analysis and through epigenetics. When I started my lab here in Vienna, one of the first things we did was developing a single-cell method for DNA methylation analysis, and then applying this as part of the Blueprint Consortium and the International Human Epigenome Consortium to the human hematopoietic lineage. So we were fact sorting together with colleagues in Cambridge from the peripheral blood of healthy individuals, these various cell types of stem and progenitor cells, And we did DNA methylation profiling, and then using machine learning, we asked whether the computer could, based on the epigenetic traces of what these cells have gone through, reconstruct what is known about the hematopoietic lineage. And that worked quite well. So we have the hematopoietic stem cell here on top, the myeloid lineage, the lymphoid lineage, with the various progenitor cell populations. So essentially, there is an epigenetic signature that we can extract by machine learning that essentially reflects how cells came to what they are. Now, for the hematopoietic lineage, we know through like 20, 30 years of painstaking work by a whole community how this developmental trajectories look like. So you can think of this as a technical validation that this approach indeed works. But then we could take that forward and apply it to areas where much less is known about the epigenetic landscape and developmental trajectories. And one very interesting case in point is Langerhans cell histiocytosis. This is a rare disease, pediatric disease, that is characterized by these lesions in the skin, but also in the skull, that can be quite damaging for these children, to the degree that some children actually die from this disease, and many more need chemotherapy treatment. And what I found striking and interesting about this disease is that it cannot quite decide whether it's cancer or an autoimmune disease. It's driven by a very classical cancer driver mutation, BRAF V600E, that you may know from skin cancer. But at the same time, it never really gets into this evolutionary selection, somatic evolution, and genome instability that characterizes most cancers. But rather, and even in some children, it eventually burns out. So it seems that there is an epigenetic role to it as well. What we did here was that we performed single-cell sequencing as well as epigenome profiling on these lesions, and initially half expecting that these are really what pathologists told us, accumulations of pretty much the same cell. But then if you look at the molecular level, you see a whole aberrant developmental hierarchy here, hiding in this rare genetic disease, with progenitor cells, with differentiation, with even a lineage choice between a more maturing and a more destructive cell type, all reconstructed based on entropy and computational methods of these single cell profiles, connected with epigenome profiles at the from the ATAC-seq profiling. So essentially, big picture emerging here, that this developmental landscape is not only what is normal development, but in many cancers and rare diseases, it means it seems to be that there are whole subsections of epigenetic developmental landscapes that are essentially taking an apparent role of development. Now, I said my title was looking into the past and future of cells, and a good place to look into the future of cells is clearly the immune system, because the whole point of our immune system is to prepare for things to come and challenges that our body faces quite suddenly when a pathogen attacks or when there is tissue damage. And the postdoc in the lab, Thomas Krausgruber, he a very experienced immunologist who had already worked on the lymphoid and the myeloid lineage, he thought it would be very interesting to look at the immunity of structural cells. Structural cells being the fibroblasts, the endothelial cells, the epithelial cells that essentially give shape to our body without necessarily contributing much to the narrowly defined function of organs. So he essentially sorted from mice these structural cells from 12 different organs of the mouse and analyzed them for immune gene activation. And what we saw was that these structural buildings of the body are no dumb Lego pieces that essentially keep us in shape, but they are really immunological cells to some degree. In a way, we know that also from non-vertebrates, that there is immunity in non-classical immune cells, but kind of the degree to which this happened also in a tissue-specific manner was quite surprising to us. Then, from an epigenetic perspective, what I found most interesting is that these cells seem to prepare epigenetically for immunologically challenges that have not happened yet. So normally when we correlate the chromatographic gene accessibility landscape with gene expression, we see a pretty decent correlation. It's not perfect, but it's pretty decent. Whereas what we saw here, that a significant number of genes, most notably including immune genes, were maintaining an open chromatin state under homeostatic conditions that was not reflected by very high levels of expression. So what seems to be happening here is that cells invest energy, effort, and also risk of things going wrong into keeping immune genes in an open chromatin state that we interpret as unrealized epigenetic potential, the ability to quickly upregulate these immune genes when a challenge comes about without the need for extensive chromatin remodeling. So essentially, even these terminally differentiated structural cells retain a level of epigenetic plasticity that allows them quite rapidly to change and adapt in response to stimuli. Now, this is a hypothesis that needs validation, and we validated it by infecting mice with a systemic infection model, the LCMV virus that infects most organs. And we indeed saw here kind of similar to a kind of an enrichment for genes that are in this epigenetically poised or high potential state that are rapidly upregulated by the virus. But what you also see that clearly doesn't apply to all genes. And that also makes a lot of sense because the structural cells need to prepare for a lot of things that could happen to them. A viral infection like we implemented here, but also bacterial infections, sterile inflammation, physical insults, wounds, metabolic changes, all of these things are things that cells need to prepare for. So to sum up this first part, the epigenome clearly gives us a way to reconstruct how cells became what they are, which is really interesting in cancer because we can reconstruct cells of origin and trajectories where we might potentially even intervene in kind of cancer stem cell type of context. But also as demonstrated here in the immunity context, the epigenome prepares cells for things to come and allows them to flexibly adapt to future challenges. And this latter thing is what we're currently trying to exploit from a more engineering perspective in order to make better cell-based therapies, to make CAR T-cell therapies that can go into solid tumors and not only survive in this kind of very hostile tumor immune microenvironment, but that are programmed to kind of thrive there and very rapidly engage tumor cells. Now, this therapeutic perspective of kind of epigenetically programmed cells, that's still a long way to go, but where epigenetics today already is having quite some impact on medicine is in the area of diagnostics, precision medicine, biomarkers. So clearly here, the epigenetics and DNA methylation in particular fills a very interesting gap between the genomic DNA sequence that is stable and informative, but doesn't really reflect any tissue-specific changes, as well as the transcriptome, which is highly fluctuating. And indeed, DNA methylation these days is the best kind of molecular marker of aging, of both chronological aging and biological aging that we have. And it's an excellent biomarker also of tissue identity that is extensively used for liquid biopsies. We first got into this topic of epigenetic biomarkers through a collaboration with Manuel Stella, studying cancers of unknown primary site, metastatic cancers, where the first diagnosis is made not in the of the original tumor, but of a metastatic lesion. And the hypothesis was that kind of epigenetically, along the lines what I just explained, these cells keep a memory of the cell type they were originally derived from. And indeed, when we trained elastic net classifiers, we could assign these cancers of unknown primary site to the most likely organs from which they were originally derived. And that was taken forward by the group of Manella Stella in a large-scale retrospective validation study that led to biomarker certification with high sensitivity and specificity, and was applied to patients that prospectively that could then be assigned to a specific therapy as opposed to empirical therapy. And there was indeed a survival advantage for those that could be assigned based on the knowledge which type of tumors these are. Now, this is not a full-blown randomized clinical trial yet, so more work needs to be done here, but it's really promising in that this epigenetic memory and epigenetic cell identity does have a value for diagnostic purposes. In Ewing sarcoma, again a pediatric disease, we see something even more interesting because it's not just organs, but it's really a differentiation trajectory that is captured in time here. So we see the kind of an epigenetic spectrum of the disease rather than clear-cut clusters, suggesting that this is kind of a stem-like cell here differentiating to a more mesenchymal cell. And during this path of differentiation, the cell is susceptible to transformation by a driving fusion oncogene, and this oncogenic driver essentially freezes the cells along the trajectory of the epigenetic landscape, keeping a memory of where these cells are originally derived from. Now, these epigenetic signatures can even be detected from blood, either based on DNA methylation profiles, or as we did it here computationally, simply based on the fragmentation patterns of the DNA that is shed from the tumor cells into the bloodstream, such that we can really use epigenetic signatures to monitor in high resolution what happens in solid tumors and also what happens organismically. So here, when these patients are treated with chemotherapy, we also see a liver signature going up because of the toxicity to the liver. So essentially from the blood and using epigenetic signatures, we can take kind of a current census of cell death in the organism. More dynamically at what happens over time, leukemia is obviously a very powerful model disease because we can take blood frequently and rapidly. So we've done a lot of work on recent years on chronic lymphocytic leukemia, very heterogeneous disease that really reflects the kind of B-cell differentiation trajectory. Again, giving kind of a case where cell of origin is frozen and maintained in the cancer cells. It's also a disease for which there is now a targeted therapy that is very effective, which is ibrutinib, inhibitor of B-cell receptor signaling that these leukemic cells are addicted to. So what we did here together with colleagues in Budapest and Hungary is that we took a relatively small number of patients, just 7 patients, and really followed them with single cell resolution and epigenetics over their immediate response to therapy. So for each of these patients, we took up to 8 time points, we said 6 different cell types, because we really wanted to see what happens dynamically on kind of day 1 after treatment, day 3 after treatment, 7 days, 1, 2 weeks, 3 weeks, etc. So really high resolution profiles. We fed that into Gaussian process models, computational models of the dynamics of what is happening here. And we could computationally reconstruct the programs that are triggered by taking these patients on therapy. And quite consistently here, we see that the moment patients go on therapy, within a day, NF-kappa B binding in these leukemic cells is being is going down, and then lineage transcription factors are shutting down, and as a result, we see an erosion of leukemic cell identity. So if you remember the epigenetic landscape from the beginning, we are not going into a differentiation block here, but we're really going off the chart with these leukemic cells. They end in somewhere, some no man's land of cell type that is not permissive normally to a cell. They do not go back to a normal cell type, but they go into this kind of aberrant cell type that they realize and go then into quiescence, which is also why the therapy works so well, because these cells essentially shut down them and just kind of linger around and do not contribute to the disease anymore. But this is also the limitation of the treatment, because the cells do not go by and large into apoptosis. They kind of hang around in different parts of the body, and they are rapidly reactivating when this kind of constant treatment with the ibrutinib is removed, which triggered us to ask if we could use chemical screening to identify drugs that might synergize with the ibrutinib. So we did chemosensitivity profiling in patient samples taken before and after the ibrutinib treatment, and identified, based on the epigenetic signatures as well as the chemosensitivity responses, what could be good combination therapies, and found some interesting candidates here. Now, this is early stage and more needs to be done, but it clearly shows how combination therapies can push cells over the brink by exploiting these induced vulnerabilities. Which brings me to a kind of summary of this part. We would really hope that we can start chronifying some of these diseases by looking into kind of the cellular responses to therapies. So rather than going in with kind of one treatment at a time, one targeted therapy at a time, that we drive to terminal resistance, we would ideally combine several drugs at a time and monitor with liquid biopsy assays constantly what is happening. And then the moment resistance emerges, take these therapies offline, go in with different therapies. Very much inspired by what has been quite a successful strategy in HIV therapy, adapting failing therapies very early on rather than driving them to kind of exhaustion and terminal resistance. Now, this is going to be much harder for cancer just because the cancer genome is so much more complex than the HIV genome, but it is giving us a very, very interesting challenge for the next 5 or 10 years, basically. In the last 5 minutes or so, I would like to go a bit more causality. So everything I presented so far is using high throughput methods with computational analysis, but has a bit of a descriptive ring to it. So if we eventually want to get to therapy, we need to kind of conclusively prove function. And that classically meant to go into kind of small scale biology, making the mouse, essentially what I call the one gene, one postdoc paradigm, that kind of you assign a postdoc to a single hypothesis of a single gene, and that is then kind of 5 years work to work that up. If you're lucky, it works out, and if you're not lucky, then that's a problem. So I would argue that leads to projects being slightly conservative often and sticking with the usual suspects for which we already know that these are important genes. And if we can now combine all the tools that have become available recently, including CRISPR screening, organoids, and computational deep learning approaches, perhaps we get to a point where we can establish causality at scale, not necessarily genome-wide, but for something like 100 genes at a time, which would give us much more confidence, including some of the long shot predictions that come from the computational models that we are running. Now, the big innovation here really is CRISPR screens, and we've just written, or I've coordinated a kind of community review on CRISPR screening that's really meant as an introduction to the topic. we in particular with the single cell community in mind, but essentially anyone who wants to kind of become more functional from a or more high throughput from a kind of more descriptive high throughput approach. And clearly the breakthrough here, in my view, was to go beyond classical survival cell proliferation CRISPR screens to marker-based screens, and in particular to single-cell CRISPR screening, where you can use single-cell sequencing as your readout and gene expression as kind of the measure of your phenotypic response, because it's almost anything that happens to cells is somehow correlated to a transcriptional signature. So if you can make the transcriptome the readout of your screen, you can screen for pretty much anything, and you do not have to make up your mind up front what you're actually screening for, because you can analyze your data against any such signature. Now, this is hard, and it took the community quite a while to combine CRISPR and single-cell sequencing. And the reason why this is hard, because the classical assays for single-cell sequencing, like Dropseq and more recently 10x genomics, they enrich polyadenylated transcripts. That is not true for the guide RNAs, which are transcribed from a polymerase III promoter and do not have this polyadenylation signal. So we really needed to find a way to get these guide RNAs visible and accessible in the single-cell sequencing profiles. And that was done in several different ways. In CropSeq, which is our method, we exploit characteristics of the vector itself and essentially put all the complexity into the vector, which allows us to stay compatible with any single-cell sequencing assays. There are also other methods, PerturbaSeq, CrispSeq, mosaic-seq, that use other solutions for the same purpose. And these methods have really become a mainstay for figuring out kind of high throughput function, but they are limited by sequencing. When we want to with at least 5,000 to 10,000 target genes, you really need to sequence a million cells, and a million cells using current 10x technology is still very, very expensive. But essentially driven by kind of this need for a cheap assay, we looked at combinatorial indexing protocols and found them to be kind of too long and too complicated to be really useful for our purposes. So we decided that we combine combinatorial index, the combinatorial indexing concept of kind of one round of plate-based indexing with the microfluidic concept of the 10x assay in such a way that we take the single cells or the cells in bulk on 384 well plates or 96 well plates, pre-barcode those, and then pump them on the 10x system with massive overloading of droplets. Normally you have kind of 0 to 1 cell inside each droplet. Here we pump 5 to 10 cells into each droplet. So essentially 100-fold overloading the system such that we can get 150,000 cells, single cell transcriptomes out of a single channel of the 10x machine, or a million cells, over a million cells from a single flow cell of the classical 10x machine. Now, that comes with slight reduction of quality because we need to, like in combinatorial indexing, we need to prefix the nuclei, and fixing is not good for nuclei or cells. But apart from that fixing, the quality really stays pretty solid, and it's definitely useful. So for any application that really needs million scale, a single cell RNA-seq, that is a very cost-effective solution. And then there's kind of two more elements to a kind of a throughput functional approach that I just briefly mentioned. And one is interpretable deep learning, and this was work by Nicholas Vitani, a postdoc in the lab, who is now an assistant professor in Salzburg, that he kind of noticed this striking similarity between kind of deep neural networks with their many layers of control and transcription regulatory networks with kind of signaling cascades. And he essentially tried to force deep learning to happen on top of classical signaling networks and gene regulatory networks. And with a few tricks and adaptations, they are quite complex from a computational type of perspective, it is actually possible to do deep learning on these gene regulatory networks directly rather than on classical deep neural networks, and we can enforce interpretability such that each edge here is really corresponding to a regulatory property, and each knot in the network is as a regulator. So we can essentially, based on prior knowledge, we can essentially adapt prior knowledge neural networks to, based on single cell data, to specific contexts and applications. And we can do so in organoids, and organoids is really what brings everything back together, because we can do all these high throughput perturbations in a system that directly reflects what's happening in the human body. So while the human cell atlas initially really focused on what is happening in ex vivo tissues, kind of an interesting component that we're really trying to push through an EU project and is to connect this in vivo context to organoid models in vitro that through single cell profiles can be aligned to what is happening in the in vivo context. So kind of with a large collaborative network here, building an organoid cell atlas that is reflecting what is happening in vivo and kind of provide a Rosetta Stone moving back and forth between perturbable models and kind of genuine in vivo relevance. This is what I think could provide the models in which this CRISPR screening and the high sequencing could take place to establish causality. I would like to conclude and acknowledge all the people that have contributed here. I forgot to mention Paul Dattlinger, who developed the CropSeq and Sci-Fi RNA-seq. I acknowledge the people throughout the talk, and I thank you very much for listening to my presentation.
Christoph, thank you very much for this very, very, very intriguing talk and the tour through many, many aspects of your work over the recent years. I guess there will be questions. I haven't seen anything in the chat so far, so I'm waiting for your raise hand function, please. Before I start with other questions, I would like to ask one by myself, if I may. So you started off by saying that in these immune cells, you saw a chromatin, open chromatin kind of memory of genes that are predisposed to be reactive after the immune challenge comes to the cells. So what do we know, or what do you think is the initial memory triggering event? So how do these cells come to know which genes they have to have in an active state? Is there some kind of training already beforehand, or how do you envisage that?
So I was not thinking about it so much as a memory of past events, but we call it kind of potential, epigenetic potential for future events. So my guess is, if developmentally those genes have never been upregulated if a structural cell has not yet seen an immunological challenge. Rather, there are transcription factors around that are doing the kind of opening up of the chromatin without ever kind of overcoming the threshold of actually kind of full-scale transcription. It could be that there is residual transcription. And I think this is something we should look a lot more in general in immunology. Baseline activity of classical response pathways could provide a way to keep those regions open and keep those cells ready to go. But that's something for which we do not have kind of very strong supportive data at this point in time.
Mm-hmm. Okay, thank you. There's a question by Maria. Maria, you can unmute yourself, please.
Hi, thank you. Thank you, Christoph. It was a great talk. It was a lot of information. I loved your sci-fi RNA-seq. I also love the name of it. I think it's great because it's so high throughput and like for you know, population studies and so on, single cell QTLs and so on. This could really be the solution to it. What I was wondering is if you load so many cells per droplet, what's the sequencing like? And you know, like, how does your saturation curve look like? Do you sequence deep enough? Do you need to sequence much deeper? And actually, let's say it like that, the price of the sequencing is still scaling with the extra number of cells that you get. How does this look like?
So the challenges when we were starting with it, we thought there were two ways why this might not work, or three ways why this might not work. The first one is that these pre-indexed cells will just break once we pump them onto the system. That was not a problem, luckily. The second one is that the droplets will just break when we pump like 5 or 10 cells into those. Luckily, also not a problem. And then the third one is what you are alluding to. Do we have enough reagents into each individual droplet? And that indeed is the case, which even more so means that this is really something we should do, because we pay 10x so much money for all these expensive reagents. And then even in those droplets that receive a cell, it seems we have far too much reagent than we actually need to make nice transcriptomes. So we can as well load more nuclei here or more cells and use those better. Now, what does this mean in terms of saturation, cell saturation and complexity? I guess the sad truth is that you cannot ever saturate a library with 1 million cells, because it's just breaking any possible budget to sequence a 1 million cell single cell library, to bringing every cell to like 20,000 reads or something. Which also means that sci-fi RNA-seq is not the method of choice if you want to go in a few hundred or a few thousand cells to 50,000 reads per cell or 100,000 reads per cell to really get into the lowly expressed genes. Rather, this is the method when you essentially do relatively shallow fingerprinting with perhaps 1,000 reads per cell or maximum 5,000 reads per cell, which will give you a lot of information. And then if you take kind of meta-cell approaches or bioinformatic approaches where you pool the reads across thousands of cells or hundreds of cells behaving in the same way, you can get very, very high resolution and high quality aggregate transcriptomes, but not for single cells, but really on the aggregate level. So I guess it's not by surprise that this is a technology that was developed by, like, we are computational people kind of by mindset, and we felt that we could profit from something like that because we have a computational way of thinking about it. If you want data with minimum computational efforts, perhaps you're still better off with the classical 10x profiling. This does come with certain computational challenges because you need to model. So when you apply this to CropSeq, then you need to do pretty advanced modeling for all of these perturbations. But to me, that makes it exciting, and it enables a type of screens at a scale that would not be financially viable without this assay.
Thanks.
Thank you. So Mark, you can unmute yourself, please, and ask a question.
Yes, thank you. I think the concept of using epigenomics to find origins, or the defining or exploiting, characterizing the history of a cell, quite exciting and has been demonstrated, for example, with the concept of cell of origin in cancers, that many people use this. What always somehow worries me is also another phenomenon that has been described quite well, like oncofetal reprogramming. So I always wonder, how can I distinguish between a cancer-depending reprogramming of a cell and the real origin? So this is something that I always need to think about, and I would be very curious to hear your thoughts about this.
I think the answer is DNA methylation and looking into the kind of repressed and not so important parts of the genome, where the cells by and large do not bother to reprogram those when they are reprogramming. We also know this from transdifferentiation. When we transdifferentiate fibroblasts to neural cells, they become quite capable as being neural cells. But if you do DNA methylation profiling in the kind of heterochromatin areas of the genome, they do retain quite a strong signature of fibroblasts. So I think of them as fibroblasts at heart with a lot of neural makeup on top. In iPS cells, it's a bit better because the iPS reprogramming process is just kind of so much deeper in its repurposing of developmental pathways. But even there, based on DNA methylation, cell of origin signatures can still be seen. And I can epigenetically distinguish an iPS cell that is derived from a fibroblast from an iPS cell that is derived from a lymphocyte, basically, or from a blood cell, because this epigenetic memory is retained to some degree. So then what this means for analyzing cell of origin is one has to look very carefully, including those kind of non-obvious regions that keep a memory of what the cells originally were.
Thank you very much.
But if we think about a technical approach to be able to distinguish between the question that Mark addressed, so what is the origin? Is it the cell of origin, or is it a kind of a reprogramming event? So that means that you need to be quite deep in your DNA methylation analysis possibilities. And where do you see this in the future, Christoph?
We absolutely need better single-cell DNA methylation assays, because strikingly, DNA methylation was I think the first epigenetic mark that was studied at the single-cell level, and it has somehow fallen out of fashion. Even after that we developed one of the early protocols, we are not really using this at the moment because it's just so tedious and low throughput. We absolutely, and the reason why this is the case is because bisulfite is so bad for cells. We really need to exploit the enzymatic approaches that can replace bisulfite for much more scalable, high throughput single-cell DNA methylation assays. And that, I think, would very much reshuffle the current situation where everyone is doing single-cell taxi just because it's easy, not necessarily because that is more meaningful than looking at the DNA methylation profiles.
Totally agree. Thank you. There's Denise who wants to ask a question. Denise, please unmute yourself.
Yeah, I just wanted if you can comment a bit on the difference between your method for high throughput CRISPR screening and perturb-seq. What's the difference, and if it's like in terms of maybe the number of gene you can profile, is it more scalable, or yeah, what does it bring on top of perturb-seq?
So obviously CRISPR and perturb-seq, they were all published around the same time. And back then, when it first started, there was very nice work by Jay Shendary lab that showed that CRISPR was superior because it did not, it avoided a level of kind of reshuffling or essentially of barcode mismatches at the moment that you're making your viruses in a pooled format. Is because if in the CRISPR, the guide RNA barcodes itself, exploiting a trick of the retrovirus that copies the guide RNA from the 3' UTR to the 5' UTR of the virus. This is why we do not need any external barcodes. We can use the guide RNA barcoding itself, whereas Perturb-seq relies on separate barcodes, and that in the original iteration of the project, of the protocol, caused some technical problems, which were discussed at length. Now, obviously, Perturb-seq has also gone through several iterations. 10x Genomics has provided support for it. That is kind of avoid some of these problems. Crop-seq has also been developed further. So I don't think there is. I think all of these methods are good methods, and all of them can be used very productively. I would still argue that Crop-seq is perhaps the method of choice when you want to go for very large screens with thousands of target genes at the same time, just because it makes it very easy to also to even do a pool screen and an arid screen with precisely the same type of setup. So you can do a few pool screens to optimize your assay, and then do the very expensive Crop-seq screen with single cell readout only once you have optimized the whole process. Certainly, we have seen a lot of uptake of Crop-seq. Like the vector is very popular on AdGene. People have a lot of people have used that protocol, and with very good results.
Okay, thank you.
Okay, so thank you very much again, Christoph, for this fabulous talk and the presentation and stunning results that you were sharing with us on many facets and levels of from basic research to applied research. So thanks again, and I thank to everybody for staying with us and listening to Christoph, and I hope to see you again on the 22nd of April. And with that, I would like to say goodbye, and yeah, thanks to everybody who organized this meeting as well. Bye. Bye, Christoph.
Thank you. Bye.